Hello again, ladies and gentlemen. We are now on our 7th lecture for the semester. The end is getting closer, and so is that practical exam. So don't dilly-dally and start reviewing histology, models, and putting together the puzzle that is the human body. This week we will be looking at the urinary system, and on your screen right now are urine samples that appear regularly in urinalyses conducted at hospital labs. This image is from Live Science, and there's a really interesting article written by Becky Oskin that goes into detail about each of these samples. The colors are the result of various substances found in urine like blood, various drugs and chemicals, bacteria, or infections. The urinary system is responsible for filtering material out of the blood through capillaries called glomeruli, helps regulate fluid volume and electrolytes. You may remember this from the endocrine system while learning about ADH, calcitonin, or parathyroid hormone. Concentrates and releases waste through the urine, and this system also reabsorbs important products back into the blood. Let's get our bearings in the abdominal cavity to be sure we know where we are. Here is that large inferior vena cava bringing blood back to the heart from the lower body. The aorta, more specifically the abdominal aorta, taking blood out to the lower regions of the body. And our first major organ of this week's system are the two kidneys. They are positioned deep to your stomach and on the left and right sides of the spine. They are just over 4 inches long, and they each have a little hat. Well, not really. That's the adrenal gland you learned about the first week. Each kidney connects to a ureter. If you remember, this is the slide you have been studying in lab when practicing your transitional epithelium. The ureter is a tube that takes urine formed in the kidneys down to the bladder. This is the storage tank for urine, which is connected to the external environment through the urethra. In this slide, you can see what structures would look like on a cadaver. The right kidney is surrounded by a lot of adipose tissue, which offers protection. Here you can see the left renal artery coming off the aorta and leading to the left kidney, which has been dissected. In lab this week, you will be dissecting kidneys, so ideally, you will want your kidney to look like this one so you can identify all of the internal structures. The two ureters come out of the hilum and lead down to the bladder. Now let's identify some of the structures on the kidney you will be finding in your dissections. The most superficial layer is the connective tissue capsule. Deep to that is the renal cortex, and deeper still is the medulla. You will learn more about these two regions and various structures that are located in each one. The renal pyramid gets its name because it looks like a pyramid, and they are separated by the renal columns. Notice that the tips of the pyramids are called renal papilla. Urine leaks out of the renal papilla and is collected by the minor calyx. Urine in the minor calyces is directed into the major calyx, which directs urine into the renal pelvis, and finally into the ureter, renal artery, and renal vein all enter or exit through the hilum. Your finished kidney dissection should look similar to this today. Here is a pyramid. At the tip of the pyramid is the renal papilla, which is surrounded by the minor calyx that converges into the major calyx, into the renal pelvis, and finally into the ureter. All right, your favorite, more blood vessels. Let's begin by identifying some blood vessels specific to the kidneys and the formation of urine. Together, the kidneys filter about 21% of cardiac output. After blood leaves the heart, it flows through the aorta, to the renal artery, into the segmental artery, to the interlobar artery, to the arcuate artery, to the interlobular artery, which branches into the afferent arteriole. That afferent arteriole brings blood to the first capillary bed called the glomerulus. You can't see the glomerulus in this image to your left, but we will look at it on the next few slides. Blood is filtered here and then leaves through the efferent arteriole. That leads into either the peritubular capillaries or the vasa recta. Both of these are the second capillary bed in the renal portal system. Various nutrients and molecules are reabsorbed in these second capillary beds. This is the third portal system of the semester. We have our hypophyseal pituitary, we have our hepatic portal, and now we have the renal portal system. Blood then begins its route back to the heart through the interlobular vein, to the arcuate vein, to the interlobar vein, then the renal vein, and finally back into the inferior vena cava and to the heart. This image is from an SEM showing you the glomerulus and some of these blood vessels we just talked about. Here is the interlobular artery and the afferent arteriole branching off to the right, leading to the glomerulus, and then over to the efferent arteriole, and finally into those paratubular capillaries. Looking a bit closer, here's the cortex of the kidney. Below that is the medulla, and they are separated by the arcuate artery and vein, so those are good landmarks to know. Remember, the arcuate artery brings blood to the interlobular arteries, which deliver blood to many smaller afferent arterioles. Those lead to the small capillary beds known as the glomeruli. Blood then moves into the efferent arterioles, which lead to either the paratubular capillaries or the vasa recta before returning back into the interlobular veins and then into the arcuate veins. I hope this zoomed-in view helps clarify and shows you the portal system a little bit more clearly. So let's focus a little bit smaller on the nephron, or the functional units of the kidney. Each kidney has over 1 million nephrons. The first structure is called the glomerular capsule. You can see another SEM of this structure in the lower left corner of your screen. It surrounds the glomerulus, the small leaky capillary bed, and acts like a catcher's mitt collecting filtrate. As fluid is filtered out of the capillaries and into the glomerular capsule, it begins the transformation into urine. The fluid then moves into the proximal convoluted tubule, so literally the twisted tube that is closest to the glomerular capsule. The fluid is transported through the nephron loop, through the distal convoluted tubule, and many distal convoluted tubules converge on collecting ducts. So now we're familiar with what a nephron looks like. What is the difference between the two nephrons on your screen? The most common type are called cortical nephrons, and they are located primarily in the cortex. The other type are called juxtamedullary nephrons. These are less common and are located between the cortex and medulla. They have a special function in helping increase the salinity of the medulla, but you will learn more about this in lecture. For the purposes of lab, just know the two types of nephrons. Remember, the main job of this system is to filter blood and produce urine. In this slide, we'll see what happens in each of the parts of the nephron in order to make that happen. Here, in the glomerular capsule, blood plasma is filtered out by blood pressure. Proteins and formed elements usually don't get filtered out. The fluid in the capsule is therefore referred to as filtrate. In the PCT, nephron loop, and DCT all modify this filtrate. Water, glucose, and electrolytes are returned to the blood, and urea is concentrated. In these regions, the fluid is referred to as tubular fluid. Once fluid enters the collecting ducts, it is called urine. Do you remember what hormone increases the water permeability of the collecting ducts and where it is made? Hopefully you said ADH, made in the hypothalamus and released through the posterior pituitary. Okay, so we're continuing to work smaller and smaller. In the next few slides, we'll see how the different sections of the nephron function to make urine. There are 3 stages in order to make urine, and we'll be looking at individual sections of this image to focus better on what each section does. In lecture, you will get more familiar with this image, but for lab purposes, you will not need to know everything that is moving in or out of the fluid, but I'm using it here for the animations to help build those connections between lab and lecture. The first stage is called glomerular filtration, which makes filtrate. For reference, here is the glomerular capsule that surrounds the glomerular capillaries. Blood flows through the afferent arteriole, through the glomerular capillaries, and out through the efferent arteriole. You can see that the efferent arteriole is slightly smaller in diameter than the afferent arteriole, which helps increase pressure in the glomerular capillaries. This pressure forces plasma and solutes out of the capillaries and into the glomerular capsule. The fluid is a plasma-like substance, but because it is in the capsular space, it is now referred to as filtrate. Essentially, glomerular filtration would be like deep cleaning your house by yourself. All the really large items like couches, tables, or appliances stay inside the house, but all the rest of your stuff gets tossed outside. This is the same for formed elements or large proteins. They stay in the blood vessels, but the plasma and its solutes, like vitamins, electrolytes, urea, or glucose, all move into the capsular space. As a quick review, glomerular filtrate is the plasma-like fluid found in the capsular space without proteins or formed elements. Here's the renal tubule coming off of the glomerular capsule. To clarify, in this image, renal tubule means the proximal convoluted tubule, nephron loop, and distal convoluted tubule. Modification of the fluid occurs in these structures, each region having its own specific function. Here are the paratubular capillaries, the second capillary bed of our renal portal system. If you remember from previous slides, the paratubular capillaries surround the renal tubule. The glomerular filtrate moves through the renal tubules, and at the same time, blood is flowing from the efferent vessel into the paratubular capillaries. In our analogy, we threw everything out of the house, so now it's time to figure out what we really need to bring back in, or if there's anything we missed that we can get rid of. The first step is called tubular reabsorption. Many people mistake this term for reabsorbing substances back into the renal tubules, but we are actually reabsorbing water, electrolytes, and other molecules filtered out in the glomerulus from the renal tubules back into the blood of the capillaries. The square moving from left to right is specific to molecules being reabsorbed in the PCT. To reiterate, different molecules are being reabsorbed in the nephron loop and DCT. Tubular secretion, on the other hand, removes certain substances that weren't filtered out in the glomerulus and moves them from the capillaries into the renal tubule. Again, different molecules depending on the location. So a quick review of this slide. Tubular fluid is located in the renal tubule, which consists of the PCT, nephron loop, and DCT. The fluid gets modified by tubular reabsorption and secretion. Reabsorption returns molecules back into the blood, while secretion removes molecules from the blood.